Okay, let's pick up where we left off. So does that look all right? So we left off, it was like the last duration was about the last slide we hit. Right. Okay. So so you guys are probably thinking, it's like, gosh, dude, are you ever gonna like turn the corner on this? I actually have. It's actually the decompression of the decongestion, which always takes a long time when it hits the chest. It takes forever to get out of there. And unfortunately, this one particularly loved to hang out on my vocal cords, and which is good. Don't want it to go deeper. I'd rather it not. So I'm okay with sounding like a frog for a little while. If you are. Okay, so and so you'll hear my voice kind of come in and out. So it'll kind of get a little froggy, and then it'll get clear again, and then go back and forth. So it's not quite teenagerish like it was last week. So it's a little bit more normal, but it still kind of fades in and out. A couple of things. First of all, before we start tearing into this, we should have a decent shot at getting close to finishing chapter 10. You notice we're almost due for an exam cycle, right? Next week. Couple of things to that. I do know that the withdrawal date is coming, right? So that's the 16th, I believe. And I know for some of you that is a date that you're keeping your eye on. A couple of things just to let you know, because I've been getting a lot of questions about this, not just from this class, from all of my classes, is about extra credit. There will be an extra credit assignment. It'll be in the form of a lab quiz, and it'll be kind of part of like your lab prep. So that's not going to come out next week. It's going to come out the week we do the lab review, which is not next week. That's a case study week, but the week after that is when that would come out. So that you can anticipate. Also, for those of you who did that sort of common syllabus quiz, when I get ready to put that into the grade book, that will also be extra credit. So those are a couple of opportunities. In terms of preparing for the exam itself, you probably noticed that our tutoring center, if you went to the 4th floor of Confluence, doesn't really have a lot in the way of biology. They have great math, right? But the one thing we have in this building is Metro's tutoring, which is in 1009. So it's like right in front of the offices. So it's just kind of like down around the corner beyond the butterfly. It'll be on the right-hand side. If you look up, you'll see a big sign hanging from the ceiling says STEM Learning Center. That's it. And that is, I work with the professor who runs that at Metro, and we work closely on a lot of different things. And she wants CCD students to use that because that is a grant-funded activity for them. And for them to get their grant renewed, it helps them get their grant renewed if they can show their grantors that it's not just Metro students that are using that CCD to your students are using that as well. Granting institutions love that sort of thing when 2 and 4 years work together that way. So they want CCD students to utilize that. Yeah. I don't know. They might. If you would just kind of poke around in there and see what's there. They got a series of people there with various schedules. They may have an online person there. I don't know. Is that a 1009? Yeah. So that's there for you. So if you're looking for tutoring, that's probably the most available to us. You'll notice that most of the tutoring that you see posted in the department is chemistry tutoring. Chemistry does a really great job. For some reason, bio tutoring, it's a little more challenging to get the tutoring to stick, because if you're a chemistry student and you're in a chemistry class, you know you live and die by your tutors, right? I mean, that's how you get through the class. So it's understood that you use your tutoring. Whereas in bio, most of our students are like, yeah, I don't need tutoring. So basically we have this nice tutoring center that's empty. That's kind of what 1009 is going through right now, because it's a bio professor who's writing that outfit. So if you want tutoring to be available, to stay available, go use it. That's the best way to make sure it's there. Even if you feel like you're comfortable, you know, tutors will oftentimes give you a different spin on something that you didn't consider and kind of help shore things up that you didn't know needed shoring up. Okay. We're talking about the cell cycle, right? We just got done talking about duration. And so this basically is our cell cycle. So you can see you've got your, your G1, that's your cellular childhood, your S phase, your G2, and then of course your mitosis. We're going to spend a little bit of time talking about mitosis, the phase of mitosis, and of course you go through cytokinesis, and then after that, basically you've got 2 new cells. Now, when you take a look at it, the breakdown of it, right, you can basically oftentimes break this down into 2 major chunks. Interphase, for instance, this is our preparation phase. So we're getting ready at this particular point to basically go through the separation. And of course, you've got your cellular childhood, your S phase, where you replicate your DNA, and then your preparation stage, which is your G2 stage, right? And a couple of things I want to point out here that are kind of in addition to things we already know about. First of all, in G2. So in G2, notice you have chromosome condensation. And here, chromosome condensation is beginning. It finishes in prophase. We'll talk about that here in just a second, but it begins here in G2. But what else is happening in G2? Well, we already know you're replicating your organelles, right? So you're replicating all the rest of your organelles. But in addition to that, one of those organelles you're replicating are your centrioles, right? Why are the centrioles important to replicate? Because those are your microtubule organizing centers, right? And so in addition to replicating your organelles that are microtubule organizing centers, you're also replicating tubulin. And what is tubulin used for? Well, yeah, transportation, but because you're building your microtubules, right? So what happens then is the idea of separation is that you're going to be moving chromosomes from one end of the cell to the other. How do you move stuff? With microtubules and motor proteins. So you're also busy making the motor proteins. We're going to be pulling these chromosomes along these microtubule tracks. So notice what's happening. You're basically making all the necessary materials to essentially build your spindle on which you're going to be moving all of your DNA. Now, in addition to that, you're also preparing your chromosomes. And how do you prepare your chromosomes? Well, first of all, you're adding a kinetic core to your chromosomes. Remember, your chromosomes at this particular point have been duplicated. There's a little bit of a centromere right there. And you're building a kinetic core with a little protein pad on either side so that you have something to grab onto. Right? You don't ever want to just grab onto naked DNA because it'll shred the DNA. So your kinetic cores are little protein pads, handles, if you will, that allows you to be able to attach your microtubules to them so that you can move them around. In addition to this, you're holding your sisters together by this protein glue, cohesin. right? So this is all preparation. You're all getting yourself ready to move. And so this is kind of what it looks like. So here you can see you've got your two sisters. You got your centromere right here, your nice little kinetochore pad, and your microtubules attached to that kinetochore. Then in between the two sisters is your cohesin protein, right? This is that protein glue that holds the sisters together. And so now you're ready. Everything is ready now. And so once you're ready, then you're ready to start moving toward mitosis. That's where we're heading. So some of these images may look familiar to you because we covered them during lab. So the first part of what we're going to see is G2 interphase. Now, G2 interphase, that's basically where this is a list of all the things happening in G2 interphase are what we listed before, right? So we got the duplication of your organelles, your centriole, your tubulin, your motor proteins, right? Also, remember, we also have DNA repair going on there. So if you had any mistakes in DNA, then you're basically repairing your DNA in G2 as well. So that's all happening in G2. Notice what you see in the cell in a G2. First of all, homogeneous DNA, right? It's not condensed yet, it's just beginning. But what you see here are nucleoli. So what does that mean? That means it's busy doing protein synthesis. What is it synthesizing? Well, tubulin, motor proteins, right? Things that are necessary for division, right? So you're getting yourself ready. So here you can see your centrioles have duplicated, but they still haven't migrated into their antipolar position. They still have to do that. So once you're done with G2, now you're ready for M phase. Now, mitosis, you basically have two different camps. You either have a 5-phase camp or you have a 4-phase camp. And there's really no wrong way. It's not like one is right and the other one's wrong. It's just a matter of your perspective, like what your emphasis is. Remember, the events of mitosis are a continuum. And essentially, it doesn't really matter where we draw the lines. The continuum still does what it does. So, for instance, if you are a 5-phase person, everybody agrees on prophase, metaphase, anaphase, and telophase. But typically, there's a group of scholars who toss in a prometaphase as a phase in between pro and meta. Now, if you are a person who says this is a phase, I can already tell where your background is. Your background is typically as a biochemist or as a cell biologist. Because what's going on during prometaphase are basically microtubule dynamics, right? You're basically building out microtubules. You're contracting the back end. So there's a lot of microtubule dynamics going on there. And as a result, all those biochemists who study microtubules basically are really interested in what's happening during prometaphase. And naturally, they create their own sort of transitional phase, prometaphase, where you have all of this microtubule dynamics going on. Now, what about the people who only do a 4-phase system? Well, those people typically leave prometaphase out. So they think in terms of prophase, metaphase, anaphase, telophase. I personally am a 4-phase person. Why? Because a 4-phase person typically has a meiotic background, right? So they did most of their cell cycle studies within the context of meiosis. And in meiosis, your view is a little bit different. You're not interested necessarily in microtubule dynamics, right? That's a biochemistry thing. What you're focused on is more like chromosomal integrity and the movement of chromosomes and the impact and the consequences of moving those chromosomes. That's what you're focused on. So you're not focused on microtubules. So with that perspective, when you're just focused on chromosomes instead of microtubules, then there's clearly 4 phases, right? And then a lot of times what will happen is a meiosis person will break prophase up into subdivisions. So like they'll break prophase up into 4 sub phases, like leptotene, diplotene, zygotene, and pacotene. So it's kind of like basically early prophase, middle prophase, late prophase, and chromosomal events associated with that, right? So it's just a different lens that you look at. And the reason why I mentioned that is because when we get to chapter 11, which is coming sooner than you think, and we're starting to talk about cell cycle and the events of meiosis, what disappears in the meiosis talk is prometaphase. And a lot of times that throws students, that confuses them, because they're like, well, wait a minute, we're talking about meiosis. There's like a prophase 1, there's a metaphase 1, an anaphase 1, and a telophase 1. But nobody ever talks about prometaphase 1 or prometaphase 2, because in meiosis, we're not really focused on the microtubules. We're focused on the chromosomes. And so that's the reason why I say you will see one of those 2 systems. Either one is wrong. But let's go ahead and take a look and see what's going on in prophase. Now remember, in order to understand meiosis well, you want to understand mitosis really well. So you want to be able to basically take a look at each one of these phases and say, okay, this is what happens in prophase, boom, boom, boom. This is what happens in anaphase, boom, 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 things like that. Okay, so you want to be able to rattle off and list the events that are happening in each one of these phases. So what are they? So first of all, in prophase, the first thing we see is chromosome condensation. So here, chromosome condensation finishes. So we started in G2, now we finished in prophase, yes. G2 is interphase. So that's before, prophase is the first phase of mitosis. So since you're condensing down, this is where those chromosomes get that little fat night crawler look to them. And you start to see a lot of space in between them. The other big thing that happens is you get spindle migration. Remember, it just duplicated in G2. But now it's migrating into its antipolar position, right? Basically what you're going for is a well-developed spindle where you have 2 antipolar centrioles with a healthy microtubule bird cage-like spindle growing out from it. Okay, so that's what's happening. So you're migrating into your antipolar position. The third one that happens is nuclear envelope breakdown. So you remove that nuclear envelope in order to get access to those chromosomes. So let's take a look at it. So here's you can see our prophase cell, right? So you can see these like little fat condensed chromosomes with a lot of white space in them. Also, if you notice, you see the absence of a nuclear envelope, so it's been broken down. And you see a nice antipolar positioning of the spindles. That's exactly what you want. So your spindle poles are migrating at this point into their antipolar position. And then essentially your nuclear envelope will break down. And then largely you've done most of what you need to do in prophase. So the next step is prometaphase. But I don't want to talk about prometaphase yet. What I want to do is I want to talk a little bit about where we're going. And then I want to talk about prometaphase as the way we get there. Okay. So from prophase, where we want to go is to metaphase. And what is metaphase? Well, metaphase is basically the alignment of your chromosomes in single file along this little fictitious line of central tendency called the metaphase plane. So anything on this side of the metaphase plate will move to that pole. Anything on this side will move to the opposite pole. So metaphase is characterized by this nicely aligned linear single file arrangement of chromosomes with the metaphase plate going down the middle of it. Now the question is, remember where we started from, right? This is where we're going. Remember where we started. We started with this. So how do you go from this, right? A random ball of gunk to this, a nicely lined up single file orderly line across the metaphase plate? Well, the way you get there is what's happening in prometaphase. So how does this work? So in prometaphase, when the two poles get their antipolar alignment, what's going to happen is they have to grab onto and attach themselves to the kinetochores. Well, how do they do that? Well, what they use is what I call like a fly fishing mechanism. Have you ever seen a fly fisherman? Some of you may be fly fishermen. I've seen movies with fly fishermen on it. That's about as close as I get to it. Right. But if you watch a fly fisherman, right, they are constantly, they're casting out and pulling back, and casting out, pulling back, right? They're kind of like doing this motion. If you watch, the river runs through it, right? Metronomic between 10 and 2, right? 10, 2, 10, 2. We call that the Methodist style, the Methodist technique. I'm not sure what that has to do with anything, but right. So this basically, you're constantly kind of pulling in, pulling back, pulling in, pulling back. Well, to the untrained eye, it kind of looks like, it's like, well, wait a minute, can't they find their location, right? I mean, are they having a problem finding where they want to sink the hook? It doesn't, do they know what they're doing? Well, what they don't realize is that fly fishing, unlike bait fishing, is a different technique, right? With bait fishing, you find a good location, maybe a shadowy little alcove or something like that, where you think the fish might be hiding. You toss, you cast out, and then you let your hook sink with the bait on there. And the idea is the fish takes the hook, right? In fly fishing, when you're casting out, you're not locating the hook. What you're doing is you're teasing the water. You're basically throwing your lure out there, which a lot of times you make yourself, right? What you're trying to do is you're trying to just touch just the top of the water, pull it back, touch the top of the water, pull it back. What you're trying to do is you're trying to get a fish under the water to think that your fly lure is a little fly skittering around on the top of the water. You're trying to trigger its attack instinct so that when you hit and you kind of tease the top of the water with your lure, the fish will jump at it, bite it, and you got it. So unlike bait fishing, fly fishing doesn't use bait. It uses your brain, which is the reason why fly fishermen oftentimes get hooked on fly fishing, because it's kind of the thinking man's fishing. You have to strategize. You have to outthink your fish and figure out ways of like trying to get them to take your fly. That's the reason why fly making and fly fishing is like almost a cult. It's like, and it's like, it's like a guarded secret, right? I mean, if you figure out how to make the perfect fly that gets you good success, it's like the CIA, right? I mean, I can tell you, but I guarantee you I will kill you, right? So fishermen are weird that way. Even to non-fishermen, which I'm not a fisherman, and I made the mistake of asking Dr. T where he went. He's an avid fisherman, and he's all like, "I'm not going to tell you." Like I care. I mean, I fish with a stick of dynamite. So so whatever. But yeah, fishermen are weird. That's because they spend too much time out there alone on the water, talking to fish. So yeah, you've got mental health problems if that's where you are. Unfortunately, that's where a lot of fishermen are. They're good at what they do, but yeah, but I'm not a fisherman. So, got that. That'll be a link. Yeah. Am I a fisherman? No, you're not. You don't even pretend to even care. Yeah, right. Extra credit question worth 50 points. Yeah. Okay, so that's just a recapitulation of that image. right? So then once you get yourself lined up, and so here's what's happening. So, right, so as you're sort of casting out in prometaphase, the way you cast out, remember, is you're basically just building tubulin on the end of your growing end, right? Your plus end of your microtubules. And so basically build your microtubule out. Now, sometimes you're not going to catch anything. And so you just basically retract it and you cast out in a different direction. You retract it, cast out in a different direction. You retract it, cast out in a different direction. But eventually what's going to happen is I'm going to cast it out and I'm going to grab onto a kinetochore. Then what I do is I wait for my sister pole to grab onto the other side. And what you do, then basically me and the sister pole, like a little tug of war game, are pulling on each other, and we're gently basically trying to align that chromosome in the center. So you're kind of pulling back and forth, back and forth. So you're kind of feeling each other's tension, and eventually you kind of slide this chromosome right down the middle. Okay. And so you're kind of aligning. And that's what's happening between the two sisters and in metaphase. Then at that point, once you get it aligned, you're in metaphase. From there, then you move into anaphase, right? Anaphase, of course, is basically where you have the splitting of the centromeres. So basically, the sisters move apart. But in order to get those sisters to move apart, that cohesion has to let go, right? With your protein glue. If that doesn't let go, then you're kind of screwed, right? So that cohesion has to let go. Keep that in mind. We're going to touch on that again later on when we talk about control of the cell cycle. But with anaphase, there's actually two different movements that are going on, right? So remember, you have two separate sets of kinetochores. You have two separate sets of microtubules. We talked about this, didn't we? Remember when we talked about the protists, we said that the protists have basically these thoroughfare, right? You have your two spindle poles, you have these thoroughfare microtubules that go through the nuclear envelope. And then on either side, they've got these little side kinetochores, or microtubules that grab onto the chromosomes. Remember that image? So you have two sets. You have one set of microtubules that are attached to the chromosomes, and that is doing what we refer to as anaphase A. So these guys will basically pull the sisters toward the poles. Okay. So these would be anaphase A here. Now there's another one, right? Because these are those thoroughfare microtubules that are connecting the two poles. So these basically push sister poles away from each other. So anaphase A is a pull, anaphase B is a push. You're doing both at the same time. So if you see what this looks like, here you can see you've got the pole that's going toward the spindle poles, right, where each of these kinetochores attached to microtubules are moving toward the spindle poles. But then you have the thoroughfare microtubules, which are pushing. This is your push off. Now, so basically what's happening is you have two different things going on. So I've got my anaphase A microtubules pulling my chromosomes toward me, but my anaphase B chromosomes are pushing away from my sister pole. So I'm doing like this, right? So I'm pushing away from my sister pole at the same time. What that does is it forces us into either corner of the cell, right? That's what that basically does. It's the process of elongating the cell, right? So that has the effect of basically pushing all of this here ultimately toward either end of the cell. This elongates the cell. And so here you can see your classic anaphase trailing edges. So here's your microtubules attached that are pulling. Your chromosome distorted. Now, once you actually pull them, then you move into telophase, right? So a couple of things are happening here. First of all, you get your spindle disassembly. So you basically break down your microtubule network. You also reform your nuclear envelope. So notice, this sounds kind of familiar, doesn't it? Kind of sounds like prophase, only in reverse. That's exactly what you're doing. You're reversing the events of prophase. And then, of course, you have your decondensation of your chromosomes. Now, last but not least, your nucleoli reappear. This is the sign of an active nucleus. So from here, you're ready for cytokinesis. Okay. So here's your telophase cell. So you can see you've got your two nuclei, and you can see your nuclear envelope is starting to reform around it. But notice what you still have in the middle. You still have a bunch of microtubules, right? Those polar microtubules, those thoroughfare microtubules are still there. They're still pushing against each other. So when you actually see it, you can actually see that leftover microtubule there. Well, think about this for a second. When we talked about binary fission with bacteria, right about in the same position, we saw something called a septum, yes? The septum was actually what? A ring, right? A contractile ring made out of what protein? Yeah, right. Yeah, Fitzy, right? So in the same area, we formed a Fitzy septal ring. And that ring was responsible for pinching the cell apart, right, during cytokinesis. Well, what do we find in animals? Do we find Fitzy? No, because eukaryotes don't have Fitzy, right? But what does Fitzy look like? I mean, but what does it resemble? There is a particular protein in eukaryotes that structurally resembled. Remember, Fitzy structurally looked like tubulin. And so what do we find in the exact same position, in the exact same process in eukaryotes? We find microtubules, which are made of what? Chapter 3. Microtubules are made of what protein? Tubulin. So notice the deductive stream of logic. We have already evidence of a Fitzy derived or a Fitzy containing septal ring in binary fission. We also know that Fitzy structurally looks like tubulin. And then to drive that home, like the alignment of its convergence of function and form, in the exact same process, cell division, in the exact same place where the septum formed in binary fission, we see microtubules made out of tubulin, which is structurally similar to Fitzy. Notice all of the parallels. When you see things that look similar to each other in different organisms, it's what's called homology. And homology typically, not always, suggests evolutionary relatedness, but not always. So then that's basically your telophase. Now it's off to cytokinesis, right? So we talked a little bit about this. We already know that animals basically will use this sort of contractile ring. In animals, there's more than just tubulin there. There's also actin filaments there as well. They'll form like an actin contractile ring. Actin is notorious for being able to contract that way. And so basically what it'll do is it'll pinch that elongated cell apart and create a cleavage furrow. So we saw that. So there's our cleavage furrow. In plant cells, we form the cell plate. Where basically we have our little Golgi-derived vesicles that sort of line up in the middle and they fuse together to create this little cell plate. Eventually they'll fuse with the cell wall to create the two different cells. So this is what cytokinesis looks like. So here's an animal, of course. You've got your contractile ring working with the tubulin, pinching this basically apart. And just to kind of drive home the visual for you, because a lot of times you don't get a chance to see what actual real cleavage furrow looks like. These are actually real cells. And that is a real cleavage furrow right there. So you can see how it looks like you took a cord and just basically put it right there and you're tightening the cord up and it's basically just kind of cutting it in half. That's exactly what's happening. This is also a cleavage furrow right there. So you can see this is about, it looks like a taffy pull. So it's about ready to pinch apart to create two new cells. So those are very distinct cleavage furrows. Was that a question? It's kind of more like pulling in. So it's just kind of like cinching your waist until you literally just like cut into two pieces. Talk about that right. So this is your cell plate. So here you can see you've got these vesicles full of plant cell wall material that fuse together. And eventually these will all fuse to create that common cell plate. And then eventually it'll fuse with the outer cell wall to create your two plant cells. So two slightly different mechanisms, but both very effective. Okay, so that's basically the gist of cell cycle. So now that we have a little bit of cell cycle under our belt, we now need to talk about control. Because believe it or not, cell division, it can be a very good thing and a very bad thing. Because if you're ready for it and if you need it, it's a good thing. But if you're not and you don't, it's a bad thing. So think about the analogy that we use with G1 when we talked about our life cycle, right? So we come to the restriction point, we want to have kids, yes or no. There's a point of no return there, right? So we said that you commit to the cell cycle, which basically means that you can't go back, right? Once you have the kid, it's like you can't unhave the kid. There's like a point of no return there, right? Well, the same thing is also true for the cell cycle. There are certain points of no return. You can't undo it, right? You can't untoast to toast here. A couple of those points of no return are replication of your genetic material. Once you replicate your genetic material, you're in it to win. There's no going back. It's like, great, you change your mind? Tough nuts, Charlie. You're going forward because now you got all this DNA laying all over the place. You got to do something with this, right? Remember the previous slide when we said that we have a very low tolerance for extra or missing DNA. So if you did DNA replication, you basically signed yourself up for cell death unless you deal with that DNA material. Another point of no return is separation. So once you actually separate out your sisters, guess what? There's no redos there. There's no retakes on that one. You screw that one up, tough nuts, live with it, right? There's life that you live and there's consequences that you pay. You never, ever relive a moment in the cell. That's a point of no return, right? So if you screw up the separation, sucks to be you, doesn't it? That's your consequence. Good luck. Wish you the best, right? It's not pleasant. It's not a nice Pollyanna world. It's a brutal world, actually. And so as a result, before you cross the threshold of these points of no return, there are systems in place to make sure that you're absolutely sure that you're ready to do this. Because once you do this, there's no going back, right? And so a lot of times I like to, I like to use the example of my kids going on a ride. They're not quite sure about it, right? They're like, oh, I can do this. I can do this. It's like, are you sure? It's like, you were afraid of it just a few minutes ago, right? No, I can do this. I can go on it. Are you sure? Because the second you get on there, I'm not coming with you, right? And not only that, but there's no stopping the ride. So if you're up there losing your intestines, well, we'll just have to do the best we can to pick them up, right? But once you make this decision, there's no changing your mind. So before you make it, let's take a moment and let's make absolutely sure that you're ready for this. And if you are, that's okay. But if you're not, then you'd better back it off because there's no going back once you start, right? Those are your points of no return. Now, obviously, this is a little bit more important than a kid taking on a ride that they're maybe not quite ready for, right? Because this is life and death. These are life and death decisions. As a matter of fact, everything in cell cycle is life and death. Here's the mantra of the cell cycle. You have two options here, and that's it. You do it right or you die. There is no forgiveness. There is no maybe. There is no, oh, maybe I can do it mostly right. No. It's do it right. Dot all your I's, cross all your T's, or die. There is no other option. So it's a severe world to live in. So basically what happens then is in order to make sure that you are absolutely ready to cross this threshold of no return, we install regulatory checkpoints. These are moments where the cell cycle is paused to make sure. This is kind of like me sitting down with the kid for a moment or two saying, listen, before you get on the ride, let's go over some things, right? Watch the other kids riding the ride. See that kid right there that pretty much just was hurling chunks the entire time? Watch that kid closely. Right? Do you want to be that kid? Right? Does that bother you? If the answer is no, then you're ready to go, right? If the answer is yes, then you probably stay here with me. So you're taking a moment or two, you're pausing things to really evaluate whether or not you're ready for this. And what's happening? Well, what's happening is typically you're checking for a couple of things. You're checking for accuracy, like DNA, for instance. And you're fixing errors. You make any errors, you got to fix them. This is your chance to fix them in your checkpoints.
So with somebody that doesn't have like heavy missing chromosomes, that
No, if you're missing a chromosome, there's like a catastrophic fail across the board. I mean, you're missing a lot of things. I mean, you're just literally almost not functioning. Because there's a lot of cues and signals that need to happen that aren't happening because you don't have the chromosome there to send them. If you're missing a chromosome? Well, typically what's on a chromosome, the reason why, like if you're talking about a miscarriage in the first trimester because of missing chromosome, the reason why you miscarry is because it's a higher level than just cell division. You can pull off cell division and be an absolute train wreck genetically. We know that. Why? Because that's what cancers are. Cancers are genetic train wrecks, but they divide just fine. So cell division isn't necessarily tethered to the integrity of your DNA. The reason why you do a miscarriage in the first trimester from a missing chromosome is because there's important developmental cues and information necessary to form your body that's missing. It's like building a puzzle and then you realize you're missing like half the pieces. Well, at some point, it's abort mission. It's like, okay, I'm not even waste my time with the rest of this because it's going to be a train wreck, and I'm not going to know what it looks like in the first place because there's just too many missing pieces. So I'm just going to throw this one in the trash. Right? That's kind of what it's like. But notice that doesn't, just because you're missing pieces, doesn't make it impossible for you to add the pieces that are there in place. That's kind of like what mitosis is doing. You can still do that. You just won't have the whole picture. So the other thing the cells are doing here is they're listening. They're listening to both internal and external signals. So let's think about this. So this is where it's kind of a little bit different of an analogy, right? Where we use the analogy of us basically deciding for ourselves whether we want to have kids or not, right? So generally speaking, the decision to have kids is an internal decision. It's like either internally you want to have kids or not, right? However, with the cell, there's an external signal as well. Now, in some countries, this is also true, like China, I'm looking at you right now, right? Where it's not just an internal signal, it's an external signal. The government is actually telling you, you have one kid, that's it. You have two, then you're going to jail for a while, right? And we're gonna like put the kid out on some hill to die or something like that. But that's an external signal. And that's kind of what cells are like. Remember, we used the example of the liver cell. And we said the liver cell is kind of looking around at the restriction point. They're basically saying, do I divide or not, right? Do I need to divide? Well, the liver cell internally is making the assessment as to whether or not it can divide, right? So if you're a mature cell, you've gone through G1, you've gotten up to the restriction point, then physically you have what it takes physically to divide. Just like if you're a teenager, you have a body that is capable of reproducing. It's not a biological question anymore, right? However, externally, you are being told what to do. And there are regulators and regulatory molecules that are constantly talking to you, saying, listen, I know you can divide, but I don't want you to divide because the liver tissue is healthy. We're looking good. We don't need more cells. So you just go do your thing. You go drop into G0 and you do your thing, but don't divide. That's an external signal telling this cell whether it needs to divide or not. It's listening to that. Okay. And so those are the sorts of decisions that are being made at these checkpoints. And of course, as soon as you answer those questions, you can clear the checkpoint. And so we have three major checkpoints that we see. The first one is our restriction point, right? This is basically where we're deciding whether or not to divide. And so a lot of our external signaling is telling us this, right? We're being told, yes, we need more cells, so you need to divide, right? But this is our G1S checkpoint. This is our restriction point. Once you commit to it, then you're going to go through S phase and G2, and then right at the end of G2 is our G2M checkpoint. And this is basically our preparation. So here now you're preparing for mitosis. And in addition to this, you're doing a lot of DNA repair. So you're pausing. It's like, oh yeah, by the way, you know all those mistakes you made in data replication? We're going to stop here for a second. We'll wait. You go ahead and clean that up. You go ahead and fix your mistakes. And we'll wait for a little while, right? It's kind of like a bus. Those bus drivers, some of them, they'll wait for a little while. But if you've been on the wrong end of catching a bus, you realize they don't wait forever, do they? They'll wait for a little while. But man, if you're not pulling it together, sorry, next bus is coming. You're going to take that one. I'm moving. Bye. Okay. So there is a limited amount of time that you'll wait at G2M. So you have to be able to fix your DNA. And that's what's happening here is DNA repair, a lot of that. And not only that, but remember, you're also checking down to make sure you have everything you need. You're checking to see if you've got sufficient motor proteins to move your DNA, that you got enough tubulin to build your microtubules with, that you've duplicated all of your organelles and that those are all ready to go. So you're making sure that all those are in place. So you're doing your final check down. It's like when you leave for vacation, right? So you kind of do that final check down, make sure, okay, did I turn the stove off? Right? Did all the lights turn off and the lights on timers, for instance? Did I make the arrangements for the dogs to get fed and watered for, whatever? So you're making all those final check downs. And then once you feel good about it, then you're ready to go. And that's basically where you go into mitosis. And then you go through prophase, prometaphase, into metaphase, and then you get to the alignment place. Here's where your next checkpoint is. It's called the spindle checkpoint. What are you checking for here? Here you're checking for attachments to kinetochores and for balanced tension. You have to make sure it's balanced. Why? Because remember, this is another one of your points of no return, isn't it? Separation is a point of no return. So you want to make sure everything looks good. Matter of fact, when you take a look at this alignment in metaphase, what you're doing is basically you're kind of pulling and tugging on these microtubules, making sure they're good and connected to the kinetochores, but you're also kind of giving them a little bit of a tug, and you're kind of checking them to make sure that the tensions between the two sister poles are balanced, right? It looks a lot like watching a truck driver secure his load. Like he's pulled up from the side of the highway, he's got a flat bed, right? And he's got this big old load on there. He's got it all tied down with straps. And so you can see this truck driver in there pulling on the straps, right? What is he checking for? He's checking to make sure that the straps are properly connected to each other, right? So you don't lose load. But he's also checking to make sure that the tension in that strap is tight enough to hold the load down, right? So he's checking for both tension and connection because he doesn't want to lose half his load on I-25. Because not only would you lose your load, and that would be a bad thing, nobody would hire you as a truck driver again, but potentially you could kill somebody, right? Depending on what it is that you're losing. Same thing's happening at the spindle checkpoint. So you're checking to make sure everything is perfect. Okay, why does everything have to be balanced? Well, let's take a look at the consequences of unbalance. Let's imagine that you have, here's your kinetochore. Let's say you have one pole that's stronger than the other. An unbalance, an imbalance in tension. So what happens here? So what's going to happen is everything is going to go toward the strong side because he's going to win the race. He's going to win the war, the tug of war, right? So what does that mean? That means you're going to get a cell over here with 2 chromosomes and then a cell over here that has nothing. So what do we know about having extra chromosomes and missing chromosomes. Is that a good idea? That's a lethal event, isn't it? So this is something that we refer to as non-disjunction. That's the reason why you have to have good balance. Oh, by the way, if you do have imbalanced segregation like this, can you have a redo? No. Sorry. Sucks, doesn't it? But you just, as cells, you're pretty much just gonna have to go die. That's it. That's, you make mistakes in this particular world and you live your consequences. That's all it is. You make bad choices and you live those consequences. There is no getting out of it. Okay? Unlike our world where we constantly get out of consequences. But we probably shouldn't get out of it. But anyway. So this is just kind of a recapitulation, but it shows you where the checkpoints are. So here you can see your G1S checkpoint where your restriction is in G1. Your G2M is right on the doorstep of the M phase, and your spindle checkpoint is right there in metaphase. And that's kind of where those are basically placed. So this helps you to kind of control and navigate to make sure that the cell cycle is actually moving along well. Okay. So when it comes to control, here's what we wanted to know. Let's just kind of take a look at research. Like research-wise, we knew that the cell cycle was cyclic in nature, right? And so what we wanted to find research-wise is we wanted to find the cell cycle protein, right? That had two functions to it. Number one is it had a cyclic pattern of expression. So we can explain the cyclic nature of the cell cycle. Number two is it had to have the function to activate the cell cycle genes. And that activation activity is driven by a type of enzyme called a kinase. Now, what is a kinase? A kinase is basically an enzyme that phosphorylates proteins. That's what it does. So here's a protein, a kinase will come in. It'll basically phosphorylate this protein. And when you phosphorylate that protein, it'll have one of two effects. It'll either turn that protein on or it'll turn it off. So basically a phosphorylation event can act like a light switch, turning a protein on or turning a protein off. And a kinase who does that phosphorylation is the protein that turns the switch. So we knew that whoever was in control of the cell cycle was a type of kinase, that basically the way it turned all of these cell division genes on was by flipping the switch with phosphorylation. So we needed two things. What we needed okay, hold on a second. Was we needed a protein whose concentration would show a cyclic pattern to it, right? For instance, if we want to take a look at a G1 regulator, we would suspect that the G1 regulator would have high concentrations during G1, and then after G1, it would crash, and it would stay low until the next G1, and then it would go high again, then crash and go low, and then it would basically, you would see it only during G1, right? So that's that cyclic expression. The other thing we wanted to see was its activity as a phosphorylase. That is to say, its ability to phosphorylate something. And we did find a kinase type of thing. We called it a CDK, a cyclin-dependent kinase. And as it turned out, the CDKs So CDK concentration, when we saw it, had continuous concentration. It was always around all the time. Well, here's the problem. This one we have the activity, but not the cyclic expression. In this one, we also found proteins that had cyclic expression. We call them cyclins. Cyclins had the cyclic expression, but not the activity. So you can see we like had half of the story in each one. What we needed was a fusion of the two together. A protein that showed activity and had cyclic expression. So as it turns out, as we did more research on this, what we found out was that cyclin by itself had no activity. CDK by itself had no activity. But when you added cyclin plus CDK together, then you got activity. So from this, it appears as though you need both cyclin and CDK. So if you superimpose the CDK on top of this, then what that means is wherever there is cyclin and CDK, then you get activity, which is like right here, and right there. But when the cyclin disappears, CDK alone does not have activity. So you need both of them together. They appear to work as a complex. Both members of the team need to be present, right? It's kind of like a yin and yang, right? You need both of them together to be able to form the whole. So this is the cell cycle again, only now you notice we've added an extra layer with these controllers, the cyclin and CDK controllers on them. So you go through G1, and for the most part, you have CDK, right? So CDC2, this is your kinase. It's a yeast. In yeast, for some reason, they call them CDC. In mammals, they call them CDK. This is the same thing. This is kinase. But notice you have inactive kinase on this particular side of the G1S checkpoint. Why? Because you don't have any of your necessary cyclin, which is a G1 cyclin. But then once you hit the checkpoint, you start making G1 cyclin. And as you make G1 cyclin, it combines with the CDK, and it allows you to clear the checkpoint. As you go through the rest of the cell cycle, you hit the G2M checkpoint. Notice inactive CDK on this side of it. Why? Because our next cyclin in play, which is a mitotic cyclin, we don't have any of that on this side. But once we hit the G2M checkpoint, we start making mitotic cyclin, and that allows us to clear the checkpoint. Why? Because it combines with this CDK. And that allows us to be able to clear the checkpoint. What about the spindle checkpoint? Well, the spindle checkpoint itself is actually cleared by a different thing called the anaphase promoting complex. So the anaphase promoting complex will actually be the one that's checking for all these connections and tensions and things like that. Once it feels like it's good, then it'll release that checkpoint and allow the cell to continue. So this is the cell cycle with the CDK in checkpoint control. Notice what's happening is we're building this up, we're layering it up, right? So we started off with just a cell cycle, then we added checkpoints to it, and now we added the CDK cyclin over it. So we got multiple layers here. So one thing about CDK, however, we noticed is in studying CDK, we noticed the CDK itself is also controlled by phosphorylation. This is a very common thing. Oftentimes, a phosphorylator is controlled itself by phosphorylation. And we see this a lot, right? So oftentimes, these proteins that you turn on with the phosphate, their job is to go turn on another protein by phosphorylating. So it's like you're turning on a light switch to turn on a light switch, but then turns on another light switch, but then turns on a light switch, and then turns on another light switch to finally turn on your lights, right? Exactly. Well, in this case, you have a fuse box of fuse boxes, right? So lots of fuse boxes in that series before you get to your final wall switch. So let's take a look at a couple things. First of all, so this CDK cyclin complex has a name. It's called mitosis promoting factor. And this basically represents your active kinase. However, when you make this, it doesn't mean that it's just all on and you're starting to phosphorylate everything all over the place like you have no control over it. You can still control it, right? Which is good. And how do you control it? Phosphorylation, right? So let's say, for instance, you make your CDK cyclin complex, but you don't want it to fire off just like randomly, like just without you controlling it, right? You want to be able to tell it what to do and how much to do and that sort of thing. You could still do that because you can actually regulate the CDK by phosphorylating it. It's got two phosphorylation sites. The red site will actually be an inactivation site. So if you phosphorylate CDK at the red site, it'll actually inhibit it. If you phosphorylate at the green site, it'll activate it. So you can turn CDK complex off and on by these phosphorylation sites. So you're controlling your controller with phosphates. And that's basically how you control the function of this mitosis promoting factor. Some cases, this phosphorylation will be an inhibitory thing. Sometimes it'll be an activator. So you don't know. It depends on the protein that you're turning on. Now, what does the anaphase promoting complex do? Well, first of all, what the anaphase promoting complex does is obviously it checks for connection. Make sure that's okay. Once it is, then what it has to do is it has to actually remove the cohesin. And it does so by making an enzyme called separase. So separase will actually destroy cohesin. That removes cohesin. Allow your sisters to separate. Now, normally, separase is inhibited. So there's a big inhibition on separase. However, when the anaphase promoting complex senses good tension and good connection, it can remove the inhibition of separase, which can then remove cohesin, which will allow you to separate your sisters. And so that's actually what the anaphase promoting complex is doing. Now, one of the things also to remember, it's never that easy. There's not just a CDK in animal cells. There's lots of CDKs in there. Now, the reason why you have lots of CDKs in there is because this gives you a lot of nuances. Think about like a keyboard. A little toy keyboard for like a toddler has a couple of keys on it, doesn't it? A couple of notes. Just something for the toddler to bang around on. But it's not like you're going to be writing a big fancy sonata on that little toy keyboard, right? Why? Because there's not enough notes, right? You probably can't even manage to get Twinkle Twinkle Little Star off of it because there's not enough notes. I'm thinking of something smaller. But yeah, you're right. If you've got an octave, you could do Twinkle Twinkle Little Star. But if you want to write the tricked out sonata, you have to have more notes, right? So what do? You get the full size piano keyboard with your black keys in it, so you can do anything you want. Notice what you did. You added more notes so that you can actually do more. That's the same thing that's happening here. You add more CDKs, so it allows you to be able to do more with your cell division. This gives you more flexibility, so you can hear more external signals, which means you can adapt to more environmental changes. So there's a lot going on here, right? This is just a recapitulation of that particular image. Now, let's take a look at some of these external signals. We're talking about these. What are they? Well, a lot of what these external signals are basically growth factors. And pretty much every cell population in your body everyone is being driven by a growth factor of some sort. A good example, your skin, right? Your skin cells are constantly dividing, replacing themselves. That mitosis is being stimulated by epidermal growth factor, EGF. Your connective tissue. So if you cut yourself and you damage your connective tissue in your skin, one of the things you have to do during wound healing is you have to lay down new connective tissue. That's done by a cell type called a fibroblast, right? Fibroblasts are stimulated to divide during wound healing by fibroblast growth factor so that you can lay down more connective tissue and fix your damaged tissues. In this case, we use the example of platelet-derived growth factor, which is kind of funny because they drop this one in here, but they don't ever really explain like what this is exactly. So a platelet is basically part of your white blood cell pantheon. So in your bone marrow, you've got both red and white blood cell development happening. And there's one particular cell in there called a megakaryocyte that has these kind of rangy sort of tentacle-like extensions that will basically break off these little cytoplasmic chunks that will break off. And these are platelets. And what do platelets do? They drive coagulation of blood. So like, for instance, if you were to damage a blood vessel, one of the first things you have to do is stop the hemorrhage. The way you do that is these platelets, which are circulating through your blood all the time, they're sticky, right? They're kind of sticky, gooey little things. And what they do is they basically glom on to the injury site and they kind of create this sort of gummy, sticky plug, kind of like a plug of chewing gum, right? And so what it does is it basically gets the bleeding to stop. It's the first step in coagulation and wound healing. But the first thing is stop hemorrhaging, because if you don't stop hemorrhaging, you're going to be dead, right? So if you don't have good coagulation and platelet count, you could actually bleed out. You could bleed to death by like some ridiculously stupid thing, like a nosebleed or just, something that wouldn't hurt anybody else. But it's like, oh yeah, I just kind of banged into the side of the cupboard and I bled to death. Wow, that was impressive. Try that one when you're sitting around swapping war stories in heaven, right? It's like, how did you go? In a blaze of glory? No, just bumped into the kitchen counter. It's like, yeah. So that's kind of what happens. So now, since you're actually bleeding all the time, and actually there's a lot of micro lacerations that are happening all the time that you don't even know about. then you constantly have to replace these platelets. So these megakaryocytes are constantly dividing, they're constantly reproducing, and they're constantly replacing. And so as a result, sending the growth signal for that is your platelet-derived growth factor. So when you take a look at your platelet-derived growth factor, it will basically tether to a receptor, a special type of receptor. It's called the receptor tyrosine kinase. This is a receptor that has a little bit of enzymatic quality to it. So here's what it looks like. So you have a receptor. There's your membrane, and your PDGF will come in here and bind to that. And then you have this kinase activity, which will basically phosphorylate protein, and then that'll create this huge cascade that's called a communications cascade. And that'll kind of get things started. So that's basically a receptor that has the ability to kickstart this entire phosphorylation cascade. And we'll take a look at a picture of that actually in just a second. But what happens if you don't get cell division right? You get cancer. Cancer is very simple. Mechanistically, it's very simple. All cancer is uncontrolled, unregulated cell growth. That's all it is. Yeah. Wait, say that again? Well, no, I don't agree with that. And the reason why is because your body spends most of its life fighting tooth and nail to stay alive. If your body knew it was going to die, it would be like, okay, I'm done. Right? I mean, just because you just sort of take your last breath doesn't necessarily mean that your body knew that when you were 5. And the reason why is because I can tell you right now, my best friend who died in a car crash at the age of 23, his body didn't know he was going to be gone at 23. His body was preparing a long, protracted fight through life until he was in his 90s. Right? Yeah, I think that the idea of your body knowing that, I think that comes to this idea of senescence and shelf life. But the thing is, nobody understands senescence. And senescence, really, if you think about it, it has a lot, it makes a lot more sense in the context of the second law of thermodynamics than anything else that people studying senescence does. I mean, are cells kind of wired to have a shelf life? Yeah. Matter of fact, the cells that are in your body right now are not the cells that you originally came in with. You've been replaced. All of your cells have been replaced, shelf life wise, multiple times over. Right? I mean, you're turning your skeleton over at least once every 10 years. Right? So there is that sense. There's a replacement plan in place in your body. Your body knows that, right? But that's not the same thing as dying. They don't. Well, mostly they don't. There are some cases where they do, but it's not a normal thing usually. So those are non-divisible. And I mean, that's true for a lot of cells. I mean, your pancreas cells aren't dividing. Your stomach cells for the most part aren't dividing. Most of your body cells, I would argue, probably not dividing, right? They're being replaced. That's not the same thing, right? You'll replace them, but you have a replacement schedule. Like, obviously, you're replacing your skin cells like, whatever, a couple of weeks. But your liver cell once a year, that's how slow that is. I mean, you're not replacing your entire liver once a year. You're like, one cell is dividing once a year. That's like a long-term thing. Your skeleton's turning over like once every decade, maybe. Right? So there's different replacement schedules associated with your body. But that doesn't necessarily mean that every cell in your body is dividing, right? I would argue most cells probably aren't dividing. And especially your neurons are the classic example of that. Exactly. Yeah. So when you have like a brain damage or something like that, and you're able to recover some function associated with it, it's not because you gained in neurons. You don't build more neurons. What happens is neuroplasticity actually has nothing to do with neurogeneration. It actually is all about rewiring. Your brain is actually, it's like physiologically, your body is a master at adapting to like a bewildering amount of insult. By the time you die, when you're old and like you're dying of natural causes, I mean, your physiology has adapted and sort of duct taped the bumper back onto the car like so many times that, I mean, it is amazing this thing can still move down the freeway. Your brain is a master of that. So if it's missing something, it basically rewires itself and reconnects itself. And that's what neuroplasticity is. A lot of times people mistake that for recovery. And that's not recovery. That's just managing dysfunction. And your brain is a master at that, at managing dysfunction. This is one of the reasons why a lot of times dementia is hard to pick up on. Because a lot of times what you do is instead of seeing like your memory loss initially, what you do is you adapt to it. Your brain adapts to it and it comes up with other routines and procedures and things like that to work around it. And then you don't recognize it anymore. And so you kind of move on down the road. But basically what that means is by the time it becomes a problem, it's been working. Your brain has been adapting to this and dealing with this for a while now. Dementia? Right now, it's really, there's nothing you can do about it. Yeah, it's just largely, it's a preparation thing is what it is. It's like, you have it, let your family know so you guys can prepare if you want to make decisions about memory care, putting them in facilities, when to do that sort of thing, what the family can expect. I mean, dementia is not easy because it alters your personality. And so, one moment you're just a little sweetie pie like you normally are. The next moment you're just F-bombing and there's like no, so it can be quite tough on families in particular, on kids and grandkids who don't understand what's going on. You have to have a very thick skin to be able to live with a family member who has dementia. I mean, that's in general, right? That's not even considering having to deal with the reality that you remember this person as this robust intellect, and now you're literally just sort of seeing them as a shadow of themselves. I mean, that's a struggle, a family struggle that I don't wish on anybody. So basically then when you're talking about cancer, all you're talking about is just uncontrolled cell growth. That's all it is. And so when you think about uncontrolled cell growth, remember, do it right or die, right? But wait a minute, there's also something else here. Let's put another one in there, shall we? Do what you're told. Or die. Got to keep the severity of it, right? So notice what I didn't say. I didn't say, oh, I'd rather you do what I tell you to do. You've got one option here. One. Do what I just told you to do or die. If you don't want to do what I told you to do, guess what? You chose. Yeah. right? So not only is it do it right or die, it's also do what I tell you to do or die. That's the way regulation is. Think about it. You can't have rogue cells because that will kill the organism. We all know that, right? A rogue cell is a cancer cell. So let's take a look at this. When we take a look at cancer, this is basically a dysregulation of body cells. That's what we're talking about here, right? And there's two different ways you can look at cancer. There's environmental agents. To disturb cell cycle control. And then there's genetic. So what are some of the environmental ones? We know some of those, right? We hear about them all the time. Yeah, you probably don't want to drink a bunch of benzene because that's been known to cause cancer in laboratory mice, right? You probably don't want to drink a lot of chemicals because they cause cancer, right? You probably don't want to breathe in a big cloud of radiation. Yeah, we know that one causes cancer. Right, those are all environmental. But what about the genetic? So basically the genetic, I think I could do this in 10 minutes. The genetic basically is wrapped around these two different types of genes. Now I have to basically kind of diagram out a gene here, first of all, for you. The first thing we're going to do is I'm going to draw what's called the coding region. This is a region that becomes protein. This is some sort of cell division gene. That's all it is. Now, generally speaking, this here is the coding region. Up here is the regulatory region. It controls how the gene is expressed. And then right about in the front is what's called the promoter. The promoter drives the gene expression. But who's controlling the promoter? Well, there's a whole bunch of control elements in this regulatory region that will either activate the promoter to drive the gene activity or will inhibit the promoter to keep it off. One type of protein that will inhibit is called a tumor suppressor. It's a protein that will sit in this regulatory region and it will actually repress and keep that gene off, that cell division gene off when it is necessary. Now, if you need that gene to turn on, then you have another protein that will come in and turn the tumor suppressor off, which will allow the gene to turn on. Okay. Now, in a tumor suppressor gene, this genetically is a recessive. Remember a recessive? You need two alleles to be able to see it. Why is it a recessive? Well, it's a recessive because what happens is we get a copy of this tumor suppressor gene from mom and one from dad, right? Now, normally through life, we are constantly being bombarded by radiation. It's just a fact of living. It's just natural radiation. And we're constantly accumulating mutations. And those mutations we accumulate are random. So there's no specific gene that's accumulated. They're just, it's like a shotgun. It's just random chance that we're getting these mutations. Generally speaking, by random chance, it'll take about 2 to 3 decades for us to pick up the first tumor suppressor mutation. So you're about 20, 30 years old when you pick that first mutation. But guess what? Because it's recessive, the other one that's still good is okay, right? We're good still. However, guess what? It takes another 20, 30 years to pick that one up. And now we lose our tumor suppressor. So guess what happened? We lost our ability to keep this cell division gene turned off. And so now it turns on. Because we lost our tumor suppressor. But notice how old we are now. 60-ish, maybe. But just because we turn this gene on doesn't necessarily mean we go, we jump right to full-blown cancer. Right? We just begin that slippery slope process where we start to move toward a cancerous state. But this is the reason why cancers typically are diseases of old people, right? Why it's incredibly rarer to find young people with cancer than it is to find older people with cancer, is because it takes a lifetime of random chance to accumulate those two mutational hits. Okay. Now, once you basically remove that tumor suppressor, then that cell division gene can turn on and can do whatever it wants. Now, a proto-oncogene is similar. So let's draw the same system. Slightly different mechanism. So there's your cell division gene. There's your promoter. Still got your tumor suppressor. Tumor suppressor is looking good. But now what happens? You accumulate a mutation that turns this thing on. So this is what's called a dominant negative usually. It turns the gene on permanently in spite of regulation. Think about it. I like to use this analogy. You're a bad toddler in a room full of toddlers. And what do I mean by bad? Here's what I mean. What you want to do is you want to kill one of your siblings. That's a pretty bad toddler. But guess what? There's somebody in a room with you. There's an adult in the room. Of course, you say, you know, I want to kill my sibling. Of course, the adult says what? No, you don't hurt your sibling. That's like the tumor suppressor telling the gene, no, you don't divide. Well, here's the thing. If I'm a bad toddler, there's two ways that I can get my way. The first one is I don't like being told no. Nobody does. So what I'm going to do is I'm going to remove the adult from the room. Okay, fine. You want to tell me no? Then the first person I'm going to kill is you. So I'm going to kill you, little Miss Adult in the room. And there will be nobody left to tell me no. And then I can do whatever I want. That is a tumor suppressor gene. Cancer cells will oftentimes intentionally go after tumor suppressor genes in an attempt to take the adults out of the room, leaving them free to do whatever they want. Now, proto-oncogene is a little bit different. In this case, the adult is still in the room. You don't kill the adult in the room, but what you basically do is you basically say, listen, I'm going to do what I want anyway. Yeah, I can see you. I can hear you telling me no. What I'm telling you is I'm not listening anymore. I'm going to go over here and I'm going to kill my sibling right in front of you. I hope you enjoy the show. Right? But I am not listening to what you're telling me. So that is a mutation that has turned that toddler into a killer, regardless of what the adult in the room is telling them. That's why it's dominant negative, right? Both of those are pretty scary, obviously, right? But a couple of things have to happen in the tumor suppressor sense, right? A lot of times, serial killers in particular, a lot of them are like recessive. They're like tumor suppressors, right? They wait for their opportunity to line up just right before they actually strike. And as long as you deprive them of that opportunity by keeping the adult in the room, then for the most part, you're safe. Right? Somebody like a school shooter is more like a proto-oncogene. It doesn't matter what kind of preparation you've done or what sorts of programs you've done or what sorts of mental health things that you've done for this individual, which apparently the Nashville person had quite a bit going on there. Right? This person is stuck in the on state and they're like, I don't care if there's a bunch of police around shooting at me. I'm taking down as many people as I can, right? That would be like a proto-oncogene. It is a dominant negative, right? So instead of sort of lurking in the shadows like a serial killer, trying not to get caught, the dominant negative is like a school shooter, right? It basically steps onto the scene and says, listen, I don't care. I don't care what society's saying. I don't care what anybody's saying. I don't care about anything. I'm just going to do this, and I'm stuck in this on pattern. Okay. So that's our witching hour. We'll continue with the tumor suppressor gene. We'll kind of wrap it up a little bit, and then we'll finish off 10 and get a good healthy start at 11. Yeah.